All right, today's topic is DNA and how we came about, the discoveries that we came about, and then the structure and replication of this molecule. So the DNA basics, this is the code used to determine what you look like. This is the cookbook. So it's the instructions on how to make you as the individual organism that you are. And your cookbook is a little bit different than anyone else's cookbook, even mom, dad, siblings. So the difference in your hair texture, the difference in coloring, all of that is in this DNA. It's called the genetic code or human genome. And we've been looking and decoding this since the 1920s. That's when we first decided that there's got to be this genetic piece out there. And we wanted to figure out what it was, how it was made, and how it determines what and who you are. The first guy that came about any of this discovery wasn't actually looking for DNA. He tripped upon it. And that's the way a lot of great scientific discoveries are made. So he was looking to cure pneumonia, to figure out why people got pneumonia, why they got so sick from it, and what they could do to prevent pneumonia. DNA was the byproduct, not the goal of his experiments. He was really looking at human health. So he looked at two types of bacteria, one which he knew caused pneumonia and killed people, and one which didn't. And he took mice, because mice are our lab rats, and he injected the disease-causing bacteria into the mice, and the mice died. No surprise there. And he took the bacteria that didn't cause disease, injected it into the mice, and the mice lived. No surprise there. He mixed these two together and injected it into a mouse. The mouse died. Well, now he has kind of a baseline. He knows what kills the mice, what allows the mice to live. Now he's going to start his experiment. And remember, he was trying to figure out how to get rid of this bacteria so that people wouldn't get sick. And one of the things he did is he took heat and he heated up the bacteria, and he took that heated bacteria, injected it into the mouse, and the mouse lived. And so what he discovered was, yes, heat will kill this bacteria. I don't know if you've ever popped a blister before, but what do you do with the needle before you pop it, right? You run it up and over a flame so that the flame will kill anything that's on that needle before you pop your blister. This is kind of where that came from, right? Heat kills bacteria. So he took this heat-killed bacteria, injected it into a mouse, the mouse lived. Here's where it got weird though. He took some of that heat-killed bacteria and put it in a vial with the non-deadly bacteria, mixed it together, I don't know why, injected it into a mouse, and the mouse died. So two things that on their own would not kill the mouse, when mixed together, did. This was kind of the first idea of transformation, the first idea that there was something that transformed the non-deadly bacteria into deadly bacteria. Anytime you see a hyperlink, guys, I want you to go into the lecture after we're done with the ed puzzle and watch that video. It really does help describe in another voice what I'm saying. So a harmless bacteria had to be somehow transformed into a deadly bacteria. We call this transformation. When this new bacteria is grown in a petri dish, it looks just like the deadly stuff. Moving on, another set of scientists, Avery, and all of his colleagues, anytime you see that et al., it means like everyone else who worked in his lab. So Avery and all of his colleagues looked at what Griffith did and said, all right, now we got to figure out the We got to figure out exactly what caused transformation. And so they took bacteria and they treated it with stuff that would break down the different components of the bacteria. So they treated it with something that would break down the cell wall. Injected into the mouse died. Took bacteria, treated it with something that would break down the ribosomes. Injected it in the mouse died. Took bacteria and treated it with something that would break down the genetic material, the nucleus, injected it in the mouse lived. Now we know that this genetic material in that nucleus is what causes the disease-causing agent. And so Avery basically first discovered DNA. Chase and Hershey went, okay, we see what you did, Avery, but we want to prove it. And this is how science works. We get a result and it's not enough. Now we got to make sure that we can get that result again and again and again and again. And so Chase and Hershey, two new scientists, looked at what Avery had done and said, we're going to prove this, but in a different way. And they took a bacteriophage, which is a virus that infects bacteria, and they treated the DNA with one type of radioactive material and the protein coat with a different type of radioactive material. Let it infect a bacteria, and they found out that it was the DNA that went into the bacteria to cause harm. All they did was confirm what Avery had already discovered, but that's okay. This is how we want science to work. We want to double check, triple check, quadruple check what we think is true. Now that we knew it was DNA, the race was on to find the structure of it. And there were two labs that were looking for the structure at the same time. Rosalind Franklin was in one of these labs. And again, there's a hyperlink here so that you can watch a video about her. She was using x-rays to try and determine the structure of this DNA. And this is what her picture looks like. It's x-ray diffraction. And by looking at this picture, they realized that it was a twisted molecule. Somehow they came up with it. Other scientists, Watson and Crick, again, hyperlink video about Watson and Crick, were able to determine the structure that we're going to learn about in a minute. They came up with the first model. They got Nobel Peace Prizes for it. Theory goes that they would have never really finalized their structure if they hadn't broken in and stolen Rosalind Franklin's work on this. And so there's a little bit of intrigue here. She didn't get credit for two reasons. One, she was dead by the time they got their Nobel Peace Prize, but two, she was a chick. And at this time period in history, chicks weren't valued that much. So what does this model look like? You have the 3D space-filled model, and then you just have the ball and stick model. What we've got is a phosphate, a sugar, and a nitrogenous base. And there are 4 different nitrogenous bases in DNA. Guanine and cytosine, and they always bond together. Thymine and adenine, and they always bond together. Making up the bonds between the phosphate and the sugars are covalent bonds, super strong bonds, which is important because you don't want, your DNA just to willy-nilly fall apart. So you want to use the strongest bonds you've got. In between the nitrogenous bases, you have hydrogen bonds, really weak bonds. And that's important because you have to open this molecule to replicate it. So every time you make a new cell, you need an exact copy of the DNA. And so you're going to replicate this DNA for the new cell. And in order to do that, you have to open these hydrogen bonds. We also have to open these hydrogen bonds in order to get the message on how to make proteins, which is what we'll learn about in our next lecture. Just another vision of it. Again, phosphate, sugar, nitrogenous base. 4 bases. Adenine always bonds with thymine. Guanine always bonds with cytosine. So molecules made up of nucleotides, adenine, guanine, cytosine, thymine, 5-carbon sugar, in this case deoxyribose, and a phosphate group. Rules to live by. A purine always bonds with a pyrimidine, and your purines are adenine and guanine, and your pyrimidines are thymine and cytosine. A with T, G with C, and there's a little math you can do here. If I say that your DNA molecule is 20% adenine, and I ask you what percent cytosine is it? So if I have 20% adenine, that means I have to have 20% thymine. That means that 40% of my DNA is made up of A's and T's. The other 60% is split between guanine and cytosine. So cytosine would have 30% of your DNA. DNA must be copied for the genes to be passed on to the next generation in animals, or for proteins to be produced, so that every cell in your body has the exact cookbook. All your cells in your body have this cookbook. The difference between a heart cell and a skin cell and a liver cell is the chapter of the cookbook they're reading, but they all have the full cookbook. When we do DNA replication, each strand is complementary. It's not exactly the same, but if on this strand I have an adenine, on this strand I'd have a thymine. Each strand can act as a template for the new strand, using our base pairing rules. The process produces 2 new strands of DNA, and in that new strand, half is the original, half is new, and that's why we call it semi-conservative. This is what it looks like. So we have our DNA, and it opens up at what's called the replication fork, and we have the leading strand and the lagging strand. The leading strand gets copied completely through base pairing using DNA polymerase. The lagging strand gets copied in little segments called Ozaki fragments, and it's because of the direction that DNA polymerase can move in. The first step in DNA replication, we unzip the DNA. So helicase moves down the DNA strand, breaking the hydrogen bonds between the nucleotides, creating templates that will be used to create the new DNA. Step 2, complementary nucleotides are attached to the template. DNA polymerase is encouraging the bonding, and it's proofreading it. It's making sure that we have very few errors. Errors do exist, and that's the basis for variation. It's why we can have so many different hair colors, is through mutations, through errors in this process. But we really try and limit those errors through proofreading with DNA polymerase. The end result is 2 identical strands of DNA, each strand containing 1 piece of the original and 1 daughter strand. Okay, lots of videos to go back and click through, plus the crash course for a review.